Hello, everyone, and welcome to this audio podcast. This audio podcast as part of a Quagmire interlude. This audio podcast as part of a Labonix interlude. This audio podcast as part of a Basil scroll. This audio podcast as part of a Storm Robotics production. In this audio podcast, in this audio podcast and this sort of intermission. It's intermission. I look at sort of chemokine receptors, chemotaxis, and T cell migration. So chemokines are multifaceted proteins initially categorized in chemotactic through chemotactic influences on a diversity of lymphocytes, and primarily they act on neutrophils, monocytes, and eosinophils. Roughly 40 retrospectively, roughly 40 chemokines or chemokine receptors have been recognized have been identified in mammals. They are a cohort of sort of structure-related molecules that allow cell migration through connections with a division of 7 membrane G protein coupled receptors, G protein coupled receptors, or GPCRs. In toward the range of chemokines, the intricacy is increased by the fact that molecules dissimilar to other cytokines have overriding functions formed by a range of cell forms, and various chemokines appear to share receptors. Chemokines are subdivided by dual chief families on the fundamentals of place on a dual N-terminal cysteine on dual N-terminal cysteine vestiges CXC and CC, and relying on if the initial 2 cysteine vestiges contain an amino acid linked to CXC or our neighboring CC. The projected chemokine categorization presently in places uses CC, CXC, XC, or CX3C, followed by R for receptor, and CX3CR1 and CX3CR1. Chemokine and chemokine receptors have been a subject of discussion, and there have been numerous papers or publications that outline the role with regard to T cell migration studies. Chemokine receptors have also been of intrigue in psychological research. In psychological research, the influence of acute psychological pressure on cellular adhesion molecules and natural killer cells have also been of intrigue in research. Crucial union molecule pairs include selectins E, L, and P, the Ig-like molecules intracellular adhesion molecule 1, vascular cell adhesion molecule 1, and beta 2 and beta 1 integrins, for example, leukocyte function associated antigen 1, LFA1, and very late antigen 4. At the same time, CXCR3 was a primary chemokine receptor investigated, which is highly induced by T cell activation. CXCR3 agglutinates 3 effective provocative CXC chemokine antagonists: interferon inducible T cell and chemoattractant ITAC, a monokine induced by gamma interferon or MIG or MIG, and interferon inducible protein 10 IP10. And these chemoattractants all provoke calcium fluctuation in activated T cells, tumor infiltrating lymphocytes. CXCR3 is found on a portion of circulating blood T cells, B cells, and natural killer cells. CXCR3 is particularly in particular has expressed a significant amount of intrigue in around renal disease, looking at renal disease and transplantation. CCR7 is also a main receptor in immune reactivity, important for accurate an accurate stance on T cell regions of lymphoid organs. Early in the immune reaction, as the microvascular endothelium gets triggered, chemokines produced by endothelial cells and subendothelial tissue all produce post-platelet launch unite to the activated tissue exterior. That draws this audio podcast interlude to a close. This audio podcast is part of a Quagmire interlude. This audio podcast is part of a Labonix interlude. This audio podcast is part of a Base on Scroll. This audio podcast as part of a Storm Labonix production.